Hi, I'll be talking on chapter 3 today, and it's going to be biological macromolecules. Now, when we talk about macromolecules, we are talking about large molecules. Macromolecules means large molecules. And what should come to your mind is your carbohydrates, your proteins, lipids, and nucleic acid. And you remember that nucleic acid is RNA and DNA, right? Good. Now, so macromolecules are large molecules. Some of them have what we call monomers and polymers. And I'll be talking about all of them. But I want you to bear it in mind that when we say macromolecules, we mean large molecules. So let's go. Now, this is polymers. Polymers, from the word poly, it tells you that it is more than one, right? From the word poly, you can tell that it is more than one. So poly here tells you that it is many, right? Then mono here tells you that it is one. Now, so when we say monomer, we are talking about the smallest unit the substance or the biological molecule can appear. So for like carbohydrates now, we have the smallest unit that it can appear. Just like you say that the cell is the functional unit of life. And so even though you have a single cell, it also has the same characteristics that Mr. A has, right? Now, that's the same way that carbohydrates also have a simplest unit. We call that monosaccharide. That is the simplest unit that carbohydrate can appear. That is a monomer. And I'm going to show you about it later. Also, proteins also have the simplest unit that they can appear, and they will still be called proteins in code. And the simplest unit they can appear is amino acid, right? So the monomer is a building block, is the smallest unit of the macromolecules that can build up the larger unit, which will be called a polymer. So if two monomers are combined together, they are going to form a dimer. If three monomers are connected together, it forms a trimer. If four monomers are connected together, it forms a tetramer. Then it continues like that. But instead of calling them each of those names, we just say that once they are more than one and two, then we call them polymers, meaning that it is many monomers. All right. Now, this is a monomer, which is the example I used the other time. This is carbohydrate. It's a monomer. And this is another monomer. This is a sugar molecule, right? If two of them are linked together, it's going to give us, so we call this a monosaccharide. I will try to write that here. Mono, M-O-N-S-A-C-H-A-R-I-D-E. Now, this is a monosaccharide. You can see this is called disaccharide because monos means one. Saccharide. Saccharide is sugar. So mono means one sugar and di means two sugars. So now this is one sugar, this is another sugar. We added it together and it forms a substance, a molecule, and the molecule is a disaccharide. Now what you will take note, okay, I'll be talking about that as we go. But let me just point out, anytime bond is formed, something will be lost. And anytime two monomers are added, it's going to lose water molecule because it is a kind of dehydration reaction. You remove water to form a bond. All right? You remove water to form a bond. And when you want to break the bond, you're going to add water. But I'll be talking about that as we go. Anytime you want to form a bond, you remove water molecule. Think about it. When you want to compress two things together, they are not going to be the same. They are going to lose some space. The space they are losing is water molecule. So anytime two monomers are going to come together, they lose some space. They lose something, and what they will be losing is water molecule. And so you can see here we have the disaccharide, and the water lost is right here. And if you want to reverse the process, you are going to add the water molecule to break the bond. So as a rule of law, before we continue, anytime you are going to form a bond, you are going to lose water molecule, one water molecule. And anytime you want to break a bond, you are going to add a water molecule. So let's go. So monomers combine with each other, forming with each other using covalent bond. So they're going to use the covalent bond anytime they are joining together. You remember last week we talked about covalent bond and ionic bond. You will see all of that on your test. They are Ruth key that you should know. Now, so in the dehydration synthesis reaction depicted here, two molecules of glucose, as you can see, two molecules of glucose will link together to form a disaccharide, as we can see here. And you know glucose means monosaccharide. All right, I'll be talking about all of that. So two monomers are going to form a disaccharide, and a water molecule is going to be lost. Right? So you can see anytime you form a bond, take it as a rule of law. Anytime you want to make a bond, you are going to lose a water molecule. Now, another thing that I pointed earlier too is this is a reverse reaction. So this reaction can go other way. It can come this way. It can come back like this. And so if you want to reverse this reaction, all you need to do is to add water molecule. If you add water molecule to this, you are going to get back your monomers separately. So this is called dehydration. Dehydration reaction is when you remove water molecule because you want to form a bond. So let's go to the other one. Water molecule is lost here and here. That's what forms the water molecule. OH plus H forms H2O. So it's a kind of dehydration reaction. Dehydration means remover of water. Dehydration, remover of water molecule. All right, so this is the same reaction showing you how water molecule is lost, right? So if you want to form that, then you go. Now, the same way we form a water molecule, a bond, two monomers form the bond, join together. Anytime two smaller units are joining together, they are forming a bond, right? Now, and I told you when you form a bond, you lose a water molecule. The same way, when you want to build a bond, when you want to break a bond, you're going to add a water molecule. All right, it's just opposite. So now this is a disaccharide. This is the bond between them. We want to lose this bond. We want to have our monomers separately. What are we going to do? We're just going to add a water molecule. So that was why I told you here that it is a reverse reaction. The reaction can go forward. When it's going forward, it is dehydration. You are removing water molecule to form a bond. When it's coming backward, it is hydrolysis. You are breaking a bond by water molecule. Lysis means break. So you are breaking a bond using water molecules. So let's see here. This is here. Here you broke this bond. You broke the bond, and all you did to break the bond is to add a water molecule. And now we have different substance here. You can see this is a monosaccharide, the glucose you had the other time, and this is another one here without any connection. So hydrolysis reaction splits polymers, this is a polymer, into monomers, right? By adding water. So again, when you want to break a bond, what do? You add a water molecule. So molecular subunits can be linked to form very large molecules. So you can form more of them, more of them, more of them. Now, anytime food is rich in large biomolecules, which is macromolecules, and polymers have to be broken down before you digest them. So if you eat proteins, it has to be broken down into its smallest form for digestion. If you eat carbohydrate, it has to be broken down to their smallest form for digestion. So let's go. Now, we are looking at carbohydrates deliberately now. The monomers for carbohydrate is the monosaccharide. That is the monomer for carbohydrates. It is the simplest sugar. Now, when we say monosaccharide is a general name, there are examples of monosaccharide. So examples of monosaccharide is going to be number 1 is glucose, number 2 is fructose, and number 3 is galactose. So I just call it FGG, or you want to call it F2G. So it means fructose, galactose, and glucose. Now they all have this same molecular formula. They all have this same molecular formula. So we say, you remember I told you about isotopes in chapter 2? And you remember I told you isotopes are elements with the same proton number but different neutron numbers, right? Now we have another word that looks like isotopes, but it is different because it is isomer. Now isomers are compounds that have the same molecular formula, which is this, but different structural formulas, right? The same molecular formula, but different structural formulas. Those are isomers. They have the same molecular formula, but different structural formula, meaning that they can be arranged different ways, even though they are the same. They have the same molecular formula, but the arrangements may be different. So for glucose, you can have different arrangement. Now look at it. This is glucose, and this is fructose. Looking at this, what is the difference? The difference is the arrangement. They both have, if you count the number of carbon, they have 6. If you count the number of hydrogen, they have 12. If you count the number of oxygen, they have 6. The only difference is in the arrangement. Look at this. If you look at this end, what do you have here? HO. What do you have here? HO is still the same. OH and HO is the same. But look at this end. You have OH here. You have CH2OH here. So the only thing that is different is just the arrangement. The same number of carbons, but different arrangement. The same number of carbons, different arrangement. Look at this. This has 6 ends, but this has 5 ends. So it's the same number of carbon if you count them, but they have different ring structure. Now, so we call them isomers. All right. Now, look at this. So a glucose, all of these three, they are the monosaccharide. Fructose, glucose, and galactose. Now, the three of them can make different combinations. You can have 2 of them combined, and it will give you a disaccharide. So let's go and see that. So this is glucose. It's a monosaccharide. This is fructose, is a monosaccharide. And they are going to combine to give us fructose, sucrose. Sucrose is the table sugar that you eat. So when glucose and fructose join together, they're going to lose a water molecule. And when they lose a water molecule, as you can see here, the water molecule will be lost. This is going to be taken away from them, and this is going to be taken away too, giving us a water molecule that is lost. And I told you again, the reaction is called dehydration. Dehydration means removal of water molecule. And what is going to be formed? The bond between them is a glycosidic bond. This bond is called a glycosidic bond, and you must remember that on your test. Now, this is a sucrose. So when glucose and fructose bond together, form a bond by losing a water molecule, they are going to give rise to sucrose, which is your table salt. Sucrose is a disaccharide. Other disaccharide that we have is, we have sucrose, we have maltose, and we have lactose. So those are the three disaccharides that we have. We have maltose, you have lactose, and you have sucrose. Like I said, sucrose is your regular table salt. Maltose is grain sugar. You see that in your malt or drinks. Then your lactose is what you see in milk. That's what you see in your milk, and that's why someone that have lactose intolerance does not take milk, because the enzyme lactase is not present in the individual. All right, so that's pretty much that about disaccharide. So I've told you about monosaccharide, disaccharide, and don't forget the glycosidic bond. I told you when a bond is going to be formed, a water molecule will be lost. But for carbohydrates, the name of the bond is glycosidic bond. I'm trying to be fast so that the video is not too long, but then if I didn't touch anything I told you, I would take you that it's something you can study by yourself. Then the last set of carbohydrates are the polysaccharides. The polysaccharide means that there are more than two. If it is one sugar, then it is monosaccharide, which are the simple sugars. So you see us calling it sometimes simple sugars. Then if it is two monosaccharides, it becomes a disaccharide, which will be lactose, maltose, and sucrose. I just call this set I call it LSM. LSM, lactose, sucrose, and maltose. And these are polysaccharides. Now, if they are polysaccharide, poly means many. So it means they have many saccharides. And what do you see here? Here you are going to see starch, which is how plants store glucose. You see starch in potatoes, right? You see it in rice. That's starch. You see it in your pasta, right? Good. Then animal also store it as glycogen. So in animal, it is glycogen. In plants, it is cellulose. In bacteria, it is peptidoglycan. And in animal and fungi, it is chitin or chitin. So those are these structures in animals, in some animals and plants. Then they are associated with energy storage in animals as glycogen and in plants as starch. So all these are examples of polysaccharides. All right, this is amylose and amylopectin, and there are two different forms of starch. Now, amylose is composed of unbranched chain. You can see it is not branched, unbranched chains of glucose monomers, and amylopectin is composed of branched chain of glucose monomers. Now, because of the way the subunits are joined, the glucose chain have helical structures. You can see the helical structure here. Yeah, just take note of that for those different strain of amylose. Now, of polysaccharide rather. Now, cellulose, which is the most abundant natural polymer. Now, cellulose is what you see in the cell wall of plants, and that's why it is very, very rigid. It is cellulose. So that makes it difficult to digest because of the cellulose that is present in it. And that's why plant is said to be strong, to have that kind of structural support. You also see it is also used to make papers and wood. Now, cellulose are made up of glucose monomers that are linked by glycosidic bond. So every time two monomers are formed, there is a glycosidic bond in between them. So if it is 4, if you have 4 monomers like you have here, then you're going to have 3 glycosidic bonds in between them. If you have 5 monomers, you're going to have 4 glycosidic bonds in between them. If you have 2 monomers, you're going to have just 1 glycosidic bond in between them. So let's go. All right, I'm just going to skip all of that. Just read that. This is something you should just look at. Insects, we already talked about that they have chitin, right? They have chitin or chitin as their exoskeleton. Now, carbohydrates gives you their source of energy, right? Yes, I know usually when we talk of energy, we think of carbohydrate alone, but you should also know that fats give you more energy than carbohydrate gives you. The only thing is this: fat only gives energy when carbohydrate is not available. If carbohydrate is available, fat will be dormant, right? Fats are only activated in the absence of carbohydrate, but it gives more energy than what carbohydrate gives. Oh, it's right here. A calorie of carbohydrate will give you 4.3 kilocal of energy, whereas 1 gram of fat is going to give you 9 kilocal of energy. So the idea is that fat gives you about 2.5 more energy than what, like 2.5% more. Not 2.5%, that would be 250% more, right? Yeah, or 2.5 more than what carbohydrate will give you. So if carbohydrate gives you 1 kilocal, fat should give you about 2.5 thereabout kilocal. So it gives you like 2.5 times more. Yeah. Now let's go to lipids. I'll be a little fast to save time. Now, fat and oil are composed of glycerol and fatty acid. I'll be talking about these carefully now. Sometimes we call them triglyceride, which means it has 1 glycerol and it has 3 fatty acids. So you can see here, this is triglyceride, and you can see this is 1 glycerol, and these are fatty acid 1, 2, 3. So 3 fatty acids attached to 1 glycerol makes it triglyceride. And how did we achieve that too? It is by dehydration reaction. Anytime a bond is going to be formed, water molecule will be lost. Don't forget that. Now, fats are insoluble in water, and like I told you, they are quite rich in energy. Because they are insoluble in water, we say they are hydrophobic. All right. Now, fatty acid that are joined to the glycerol can either be saturated, they can be unsaturated, and they can be trans fats. What do we mean by saturated or unsaturated? It is simple, like you can see here. This is stearic acid and this is cis-oleic acid. What is the difference between this and this? This stearic acid has more hydrogen molecules than oleic acid. And how does that happen? They will have the same number of carbon, but they have lesser number of hydrogen. Why? Because of the presence of the double bond here. So the more double bond, look at it. This has 1, 2 double bonds. Oleic acid have 2 double bonds. Stearic acid have just 1 double bond. Now, don't care about the names now. I only care about the explanation, knowing that saturated and unsaturated. Now, the more of the double bonds you have, the less saturated the molecule becomes. And it doesn't apply to fat alone, it applies to every molecule. More double bond, less saturation. So if there's a molecule or a compound that doesn't have a double bond at all, it's going to be more saturated. but the one that have double bond is going to be less saturated. All right, so this is saturated because it has just one double bond, which is okay because this is the functional unit, right? And it is unsaturated because it has double bond here. It's functional unit as well, but then it has two instead of one. And obviously this is what we call trans fat. And I may talk about that later as we go. Let's go. Now, saturated and unsaturated fat. This is stearic acid is a common saturated fatty acid, and oleic acid is a common unsaturated fat. Now, when you talk about trans fat, you have margarine, which contain trans fatty acid. Please read this up so that I can rush up. Okay, let me also point that saturated fats are, because of the way they are tightly packed. You can see everywhere is busy. They are tightly packed, and so they are solid at room temperature. That's what you see in animals. Those are animal fats. They are solid at room temperature, and so if you have oil that are made from animal sources, it will be solid at room temperature. But that's what you see also in your meat. Sometimes the hard fats, you see that it is solid at room temperature. But this fat, oleic acid, you can see that is not as packed as this. You can see the space that should have been filled with hydrogen. So it is not as packed as the saturated one. That's why we say it is unsaturated. And it is liquid at room temperature. So the digestion, this digests faster than this. And that's why we say that plant sources of oil are usually more healthy to eat than animal sources. All right, so you see, like trans fat, you see it more in processed food, peanut butter. You see it more in like fast food, or vegetable shortening, some margarine, right, that you have here. Coffee creamer, you see trans fat there. Now, trans fat, now there's a little controversy about trans fat and saturated fat, about which one is more dangerous or not, which one is more unhealthy. But you know, trans fat adds to your cholesterol, which fairly is good, but when it's becoming too much too, it's not good. So it's usually wise to avoid like more of trans fat sources. It's always advisable. Now, they are produced from vegetable fat by injecting into liquid oils to solidify them. So they are not as healthy too, and you can see here they are usually found in processed food more. Omega fatty acid, please just read this up. It's I don't want to take more time on that. Read waxes as well. All right, like I said, some few things I don't cover is not because it's less important. It's simply because I feel you can just study them instead of making this too long. Now, phospholipids are major constituent of the plasma membrane. From the plasma membrane is usually called phospholipid bilayer, meaning that two layers of phospholipid. And phospholipid means that there is phosphorus head and there's fatty acid tail. It is more like what you have here. This is how the phospholipid is. So this is the phosphate head, and these are the two fatty acid tail. So each of them is like this. Then we have a long serial of these, and it is duplicated. So this is one layer, then another layer is going to be like opposite this. So we call it phospholipid bilayer. What is the major thing about it? It consists of fatty acid chain that is attached to a glycerol backbone. Instead of 3 fatty acids in this case, it has instead of 3 fatty acids, these are 2 fatty acids attached to the phosphate head. And the phosphate group needs to be modified by an alcohol, choline or serine. So this is what you have. You have the phosphate head, right? Then you have the 2 fatty acids here. Now, this path is hydrophilic, meaning that this path is friendly with water. It can mix with water, but this path is hydrophobic. So we call it hydrophilic head and hydrophobic tails. All right. Now, a phospholipidic molecule with 2 fatty acids and a modified phosphate group attached to a glycerol backbone. The phosphate group may be modified by the addition of charged or polar groups. 2 chemical groups that may modify the phosphate group will be choline or serine, and that is all right. So as both serine and choline, these are amino acids, right, attached to the phosphate group at a position labeled R. So you can see, so they can attach here just for modification. But majorly, when we talk about phospholipid bilayer, we are talking about the phosphate edge, which is hydrophilic, and the hydrophobic tail. So you can see you have the fatty acid, the lipid here, and that's part of why your cell membrane have its typical features. You know, the cell membrane is selectively permeable. It's because of the orientation. Okay, very good. Now look at this. This is what I was talking about. This is the orientation of the cell membrane. This is one phospholipid here. This is one. This is another. And so it forms, this is a phospholipid layer. This is a phospholipid layer. This is another phospholipid layer. And because we now have 2 layers, we call it phospholipid bilayer. Now, the tail is hydrophobic, meaning that it doesn't allow water molecule. It doesn't mix with water molecule. And that's why it is difficult for water molecule to just pass through the cell membrane, because even though water molecule can get here, but it can't get here, it will burn. It's more like you pour water in a fried oil on the stove. It's going to explode. That's what happens here. So the phospholipid layer is a major component of all cellular membranes, and the phospholipid head groups, the hydrophilic head, which is the phosphate group of the phospholipid, face the aqueous solution, right? You can see. So this is what you have on the outside, meaning that it can relate with water and it won't be any problem. And this can also relate with water on the inside and it won't be a problem. Now the hydrophobic tails are sequestered in the middle of the bilayer. You can see they can be here. So if I need to move water molecule into the cell membrane, then we use the channel which is called aquaporine. We use the protein which is aquaporine. Aquaporines allow water molecules. It allows water molecules to move into the cell, but it only allows water molecules alone. It's a transport protein that only allows water molecules to move into the cell. All right, let's go. Now these are steroids, and when you talk of steroids, the most common steroid is cholesterol, and it's synthesized mainly by the liver, and it's a precursor for many steroid hormones. Now, majorly, the hormones in your body are mostly proteins, but we also have steroid hormone. We have steroid hormone, and some of them will be testosterone and estradiol. So, cholesterol is the major precursor for all of these hormones. This is cholesterol. This is cholesterol here. All right, now let's get to proteins. Now proteins are, I told you, I can't remember if I've mentioned it, but the major function. Now, carbohydrate gives you energy, but protein does the work. No protein, no work is done in the body. That's why when your temperature is about to rise beyond 104, they ask you to run to the hospital, to the emergency. The reason is because Proteins has a structure. If you have done A and P, or you are doing A and P now, you will know that your professor will always say form relates to function. Form relates to function. Structure relates to function. No structure, no form. If you lose a structure, a specific structure of an organ in your body, the organ is going to lose its function. When the heart enlarges, it loses its function. When prostate enlarges, it loses its function. So, function relates, structure relates with function. No structure, no function. That's the same way the protein works. Protein has to have a specific structure before it can perform its specific function. No structure, no function. So one of the major things that affect the structure of a protein is temperature. When the temperature goes beyond normal, it will break the protein. We say it will change the nature of the protein. So we call that denature. Once the protein structure is denatured, if a protein structure is denatured, or if a protein is denatured, right, the protein cannot perform its function efficiently at that time. So that's why if your temperature is going higher than normal, you're advised to run to the emergencies, because proteins are very important. Now, carbohydrate gives you the energy. Protein does the work. That's where I was going. So protein is quite important. If you have energy and there's nothing to do the work, nothing is going to be done. You think about it, energy, but the machinery that does the work is protein. Now, protein is one of the most abundant organic molecule in the living system. 50% of dry weight of most cells. That's what protein covers. Now, it has several functions. Like I said, it does metabolism. So no protein, no digestion. It does transportation. It does defense. It does regulation and motion. Now, types of protein, we have digestive, enzymes, we have transport, all of that. Now, these are the examples and these are the functions. I'm not going to be reading that. Just read through that, and feel free to send me emails if you have questions. Now, what am I talking more about here is, I'm going to say something about lipid once I finish everything about proteins and nucleic acid. Now, protein also have a monomer, but you remember I didn't mention anything about monomer in lipids. I'm going to come back to that. Protein also have a monomer, and the monomer is amino acid. Don't forget, carbohydrates have a monomer, and the monomer is monosaccharide. Then the monomer can build up to two monomers. Adding together, it forms a disaccharide. And that saccharide, when it is more than two monomers, it's going to give you polysaccharide, and it continues. So protein also have a monomer, and the monomer is amino acid. Now, this is the general structure of amino acid. This R here is R group. It can be anything. We call it a side chain, so it can be as simple as H, which will be in glycine. Can be as simple as H2, can be like CH3, CH2, it can go on like that. All right, so now this is one amino acid. This is another amino acid. Basic structure of an amino acid is that there will be a central H, as you can see here, a central C carbon, as you can see here. There's going to be an amino group. It will be present. It must be there. And there will be a carboxyl group. Then you have H here. Then this can vary. But these three components must be there before you can call it an amino acid. Let me change this to a yellow pen and use it. Now these three components must be there before it will be called an amino acid. This is what varies. The R here can vary. Now let's go. So again, these three components must be present, but these can vary. Now, these are two amino acids coming together to form a dimer. That makes sense to you, right? Two monomers wanting to form a dimer. Now, but what is going to happen again? It's also a dehydration reaction. So water molecule is going to be lost anytime a bond is going to be formed. And this is the bond that is formed. Now, in the case of protein, what is the name of the bond? Are we going to call it glycosidic bond like we did in carbohydrate? No, we are going to call it peptide bond. So the name of the bond that is formed when two monomers join together in protein is called a peptide bond. Let's go. So it's a peptide bond. So when two monomers form a bond, the bond in protein is a peptide bond. And so a peptide, this is a peptide. This dimer is called a peptide. The name of the bond formed between them is a peptide bond. All right. When we have more than, okay, I don't have an image of it here, but when we have more than two forming, we call it polypeptide chain, right? If it is more than two, we call it polypeptide chain, but if it is just two, it's a peptide. If it is more than two, it becomes a polypeptide chain as it continues. Now, the shape of a protein is necessary for its function. Can you see that? Form of a protein or shape of a protein is very important to its function. Now, proteins have many shapes and molecular weights. We have globular proteins such as hemoglobin. We have fibrous such as collagen, which is found in the skin. Now, changes in temperature, you can see this is key. In pH, very important, and exposure to chemicals, they can denature, you can see that, a protein. Once it denatures a protein, it is not reversible. The protein loses its function, it loses its shape. You can see this is normal protein. Now, once it's denatured, it loses its shape, and the protein becomes irrelevant at that time. No function at that time. Now, these are lists of amino acids that we use in making more proteins. So these are amino acids, like I told you, these are the basic basic side of every amino acid that must be present. You will notice that all these are the same. It has a carboxylic group. It may look like it's arranged different way, but it's still the same. It has a carboxylic group, it has the amino group, it has a hydrogen, it has a carbon at the center. Then these can be different. You can see these can be different. All right. So those are amino acids. So now, before I continue, the monomer for protein is amino acid. The bond formed between two amino acids to make a peptide is called peptide bond. And when the peptide is becoming longer, it becomes a polypeptide chain. All right, so for the hemoglobin, when we talk about sickle cell, it's just a mutation in a single amino acid. Just a mutation. It's just that an amino acid was substituted, like glutamine. Just an amino acid was substituted in that. So the amino acid in normal hemoglobin, what do you have? You have glutamine in position 7. But in a sickle cell, you have valine replacing glutamine, and that is the difference. And that affected the whole of the hemoglobin, and it leads to a sickle cell. So that tells you how how proteins are very important with their structure determining their function. So the shape of a protein is necessary again for their function. Look at this. Levels of protein, we have the primary level, and the primary level is just a sequence of amino acids that you can see here, so we can call it a polypeptide chain. You can see here is just a chain of amino acid, and in between the amino acid you have a bond, and the name of that bond is peptide bond. Then the secondary protein is going to be, it has two shapes. Number one is the pleated shape, which is just a linkage of polypeptide chain, but they have hydrogen bonds in between them, and you have the alpha helix. So sometimes we call it the beta sheet or the pleated sheet. So is either beta sheet, beta as BETA, or the alpha helix. So we have two of them for this case. Now, what is the major? The hydrogen bond here makes them to, it has the backbone that causes the amino acid to fold into like different sheets or different pages like you have here. They are not still as functional at their level. This is not a functional protein. It's not a functional protein. This is also not as functional protein. Now, the third proteins that we have is the tertiary protein. Now, it's a 3D shape protein, but it is just for one polypeptide. So think about this polypeptide here forming this shape here. Now, that is a tertiary protein. So it has a mixture of the pleated sheet and the alpha helix all together. You can see the pleated sheet here, and you can see the alpha helix here. So at this point, what you will notice is that the difference between all the proteins is that they are changing shape. And when you want to say they are changing shape, this looks like a straight chain. This is becoming folded. This folds more, and this folds more. Now, the quaternary protein is a combination of it's an arrangement of two or more polypeptides in protein. So you can see you have this. So this would be like different arrangement of polypeptides of protein. So it will have more than one chain, right? It just has that. But what you will take note is 3D formation begins here. So these proteins are functional at this level. And when there's temperature, what is going to happen is that they will lose their folding, they will lose their coiling, and obviously they will lose their function. So take note of the the four types of proteins, be able to differentiate between them. Study it more in your textbook. Okay, so it's still the same. I don't want to take more time on that. It's becoming longer. That's why I'm not touching some stuff again. So good, I'm going to end here now. This is the last one, and this is nucleic acid. Now, what is nucleic acid? That is your DNA and your RNA. Usually, this is what we don't seem to be aware of, but we know DNA and RNA. We just don't know that they are called nucleic acid. So DNA and RNA makes up the nucleic acid. Now, DNA stores coded information. Like I said, they are DNA and RNA. So let's go. The monomer for this is called nucleotide. So two or more nucleotide is going to give you a dinucleotide. Two nucleotides is going to give you a dinucleotide, and it continues and give you a nucleic acid, right? Now, DNA, the name is deoxyribonucleic acid. So it's DNA. And RNA is ribonucleic acid. It is RNA. Now, what is the difference between RNA and DNA? The name ribonucleic acid, RNA, and deoxy. Now, when you say deoxy, it's from the word, it's like the word deactivate or remover. So it is remover of oxygen. So oxygen is removed here, and the removal of oxygen from a nucleic acid, right? So there is a sugar that has oxygen, the other sugar does not have oxygen. So that's why we call it the deoxyribonucleic acid. So the ribose sugar in DNA does not have oxygen on position 2, the carbon in position 2. So that makes it a carbon a ribose sugar. They both have ribose sugar, but DNA have a ribose sugar that is short of one oxygen, whereas RNA have all the oxygen complete. Does that make sense? And that is on carbon 2. Now, so the genes are in segment of DNA, mRNA. I'll still teach genetics as part of this chapter, so I'm not going to go more into this. Now, this is double-stranded, basically you should know, and this is single-stranded. Now, another thing I would want to tell you here that I don't have on the slide is DNA consists of some bases, and the name of the bases is cytosine. So DNA have cytosine C, it has guanine G, it has thymine, and it has adenine. Whereas RNA has cytosine, it has guanine, it has adenine, but it doesn't have thymine. Instead of thymine, it has uracil. So when you look at these ends, you will see that these are the 5 bases that we have. We have the cytosine, we have the thymine, we have the uracil. Uracil is only in RNA and thymine is only in DNA. So anytime I see T in DNA, I'm going to substitute it with U because T should not be in RNA. So anytime I see T in DNA, if I'm using that same DNA strand to make an RNA, then it will be replaced by uracil. All right, so every DNA or every RNA, every nucleotide, I don't want to go to DNA now, will consist of what we have here. It's going to consist of a phosphate group. Every nucleotide will consist of a phosphate group, number 1. Number 2, it will consist of a sugar, is a 5-carbon sugar. And number 3 is going to consist of a nitrogenous base. These are the nitrogenous bases here. We call it nitrogenous base. So now, if I'm going to ask you what makes up a nucleotide, nucleotides, first, they are monomers of nucleic acid. And what makes it up? Number 1, phosphate group. Number 2, nitrogenous base. And number 3, 5-carbon compound. Those 3 things makes up one nucleotide. Now, as an addition that I don't want you to forget, when we are talking of DNA, generally it is nucleotide, but when we are talking of DNA, we'll be talking about I'm going to write it here, we'll be talking about deoxyribonucleotide. So if I'm specific about DNA, then it's going to be deoxyribonucleotide. Does that make sense? Yes. Then if I'm talking about RNA, it's going to just be ribonucleotide. So you just write ribo, then you put nucleotide to it. It won't have deoxy. So generally, their monomers are generally called nucleotide. But if I want to be specific, for DNA it is deoxyribonucleotide, but for RNA it's ribonucleotide. And what's going to be different? So if I come here now, the difference is going to be that on this carbon 2 here, they will both have phosphate group, yes. They will both have nitrogenous base, yes. But first, you know their nitrogenous base for RNA is going to be uracil, adenine, guanine, and cytosine. It cannot be thymine. Then for DNA, you know this will be either cytosine, guanine, thymine, and adenine. But when you get to this point, they are also not going to have the same ribose sugar. It's both the same, they both have ribose sugar, like 5-carbon sugar, but on this carbon 2, RNA is going to have OH here, but DNA will only have H here. And that's the difference. All right. Now, if you take note, I told you about... All right. So, now you should note this. the monomers for DNA or an RNA, they are nucleotides, right? For DNA is deoxyribonucleotide, for RNA is ribonucleotide. The same concept, if they want to come together, it will be dehydration reaction and a bond will be formed. The name of the bond that will be formed is either called an ester bond, you call it an ester bond, or I'm writing it here, diester. Or phosphodiester bond. So that's a bond. Now if we want to make a series, you remember that on carbohydrate the monomer is monosaccharide, the bond is glycosidic bond. For proteins, the monomer is amino acid, the bond is peptide bond. For nucleic acid, the monomer is nucleotide, the bond is phosphodiester bond or ester bond. Now you remember we didn't talk about the monomer in lipids. We didn't talk about the monomer in lipids. Why? Lipids, they don't have monomers. And this is what we mean. Lipids are made up of smaller molecules, but they are not made up of related smaller molecules. Now look at this. This is lipid and this is a glycerol. Glycerol and fatty acid are two entirely different classes. They are not the same. They are not associated. They cannot be grouped in the same group. Now for glucose, galactose, and fructose, they are different, but they are all monosaccharides. But glycerol is glycerol, fatty acid is fatty acid. They are not in the same group, so we can't call them monomers. For glucose, fructose, and galactose, even though they have different shape, but we know they are all classified as monosaccharide, but not the same for glycerol and fatty acid. So because of that, we acknowledge lipid to be a macromolecule, but it is not a polymer because it doesn't have monomers. Now, monomers is just different from different smaller molecules coming together. It means they are smaller molecules coming together to form a structure together. It's not different, entirely different molecules that cannot be classified together. All right. So that is the difference between lipid and the other macromolecule. But then it is still a macromolecule. But we are not talking about its monomers because it's made up of different structures. All right. So I'm going to see you again in chapter 4. Till then, if you have any questions, feel free to reach out to me. Have a good one.